Hey guys, we're in the lab today running some experiments. We're going to do some gel electrophoresis. So yeah, first step is to make some gel.
Yeah, so I put 0.2 grams agarose. Yeah, it's going to be 1% agarose gel.
We take the agarose, we take a specific buffer, we mix it in a vial, put that vial into the microwave, a certain reaction occurs, and then we can put it together in a mold.
So I will pour this over here to make jello. Put it in electrophoresis. Apply voltage, and the samples should migrate. And then I'm going to mix it with this molecule, which is the positively charged analog of potential alien genetic polymer. So if it's a positively charged molecule, it will move.
Ooh, pretty. So what have we just done, Jan?
We run electrophoresis. The red is always plus. This is a negative charge. And we see that from the start, our compounds move towards a negative electrode, which means that it's positively charged. Daniel Hutter made actually this molecule. The orange part is this dipsil, and the backbone, which carries the positive charges on these protonated amines. So it works. We are making analog of polyelectrolyte with a sort of base molecule with a positive charge on the backbone. So that's nice. It works.
But this is the paper we're looking at today. This is written by Stephen Benner and Daniel Hutter, talking about phosphates and different genetic molecules. I have in front of me Dr. Stephen Benner.
Yes.
Okay, so we just did some gel electrophoresis, basically moving ions in the gel, the agarose gel, in different directions. Yeah, this one. Okay. In that figure, in figure 2, you're showing DNA, which has those phosphates, right? Those negative charges that go up and down the backbone.
Okay, so there's DNA, right? So you're looking at its building blocks. This is the archetypal double helix, and it contains linking phosphate groups, which are these white things going up the backbone. Those are holding together sugars, which are these red pentagons, and on the sugars are the bases.
The relevant part of the experiment that we're doing today is to show that charges with, instead of these phosphates being negative, they're actually a positive charge. They can still be moved in an electrophoresis gel.
The synthetic biologists in us are trying to make alternative forms of DNA to ask, what if and why not questions about alternative molecules that might also support Darwinian evolution. We tried to replace the phosphates, and every time we tried to replace the phosphates with anything that was neutral, the system collapsed. It could no longer handle genetic information. It could no longer evolve. And that is leading to the polyelectrolyte theory of the gene. Now, what you're seeing in this particular figure that you pulled out of this paper is where we've replaced the negatively charged molecules by a neutral linking group. Okay, so this is a dimethylene sulfone. Daniel Hutter was very much instrumental in synthesizing these. And the result was a molecule that failed to perform as an informational molecule, even for storage of information, certainly for the replication of information. And this is what led to the polyelectrolyte theory of the gene, which is saying that these repeating white parts in the backbone have to have charges on them. And in your natural DNA or RNA, those charges are negative. So the theory is that Darwinian evolution is the archetypal feature of life. That is, it's universal. It's the only way that chemicals can self-organize to give us properties that we value in life, especially the replication with mutations or replication with mistakes, where the mistakes are themselves replicable. But the idea is that a repeating charge is important for the molecule to support the informational needs of Darwinian evolution. That repeating charge can be negative, that repeating charge can be positive, but it is impossible to have Darwinian evolution without a polymer with a charge one way or the other.
So to summarize, we did some gel electrophoresis. We input some solution that had a positively charged polycation as the backbone to simulate basically an alien version of a DNA-like molecule. As Dr. Benner just talked about, we have negative charges in the form of phosphates linking the backbone of our DNA, but it's feasible that it could be positive. So we tested in the gel electrophoresis with the positive linking molecule to down the line use that same type of molecule, that same cation, in the Agnostic Life Finder ALFA device, and to test the feasibility of the polyelectrolyte theory of the gene in an experimental fashion. Thanks for coming along with us today, guys.